Okay, so last week we finished up basically all of our Mendelian processes. We talked about the law of segregation. Somebody remind me what the law of segregation tells us. Levi.
Isn't that genes are inherited independently?
Well, so that's the law of independent assortment. You're talking about that the inheritance of one gene does not impact the inheritance of a second gene. It's the law of independent assortment. And we know, because we understand how meiosis works, how that happens, right? It's either by crossing over if they're on the same chromosome, or it's by independent assortment if they're on separate chromosomes. But the law of segregation, it's a different animal. Yeah, Allison?
Is that the segregation between dominant and recessive?
Yeah, or between two copies of the dominant or two copies of the recessive, if that's what you have. But it basically tells us that when you're making gametes, each gamete only gets one copy of that factor, only gets one copy of that gene, okay? That they segregate during gamete formation. Now, we know from meiosis when that happens, right? We know from meiosis when that segregation happens, and when does it happen? When does your one copy of that gene separate from your other copy of that gene? It's anaphase. Is it anaphase 1 or is it anaphase 2? And the answer is it depends on if crossing over put the other allele with the other one or not, right? Because if it did, then it happens at anaphase 2 when the sister chromatids split. If crossing over didn't mix them at that site, then it's at anaphase 1. Okay, so today we need to talk about things beyond Mendelian inheritance. How does it get a little bit more complicated? First question we need to address is this. How does incomplete dominance compare with codominance? So we have times where it's not so simple. It's not dominant allele, recessive allele. It's more complicated how the two alleles contribute to the phenotype, okay? And so let's look at this. Here we have incomplete dominance. So we set this up just like we set up our one character crosses when we were going to do our monohybrid cross, right? Where we take a true breeding parent for one of our two alleles. We take a true breeding parent for the second allele, and we cross them. And we know that if you take a true breeding parent with the dominant allele, and you cross them with a true breeding parent with the recessive allele, in your F1 generation, you're going to lose that recessive trait, right? Right? Because in F1, what is every genotype, every individual's genotype going to be in F1? Heterozygous, which will give them the dominant phenotype, right? But look at this. Here's incomplete dominance. Our recessive trait did not disappear. And it did not disappear because this trait here is not completely dominant. And this is just a wonderful example of it, probably the best example we have in Snapdragons, where you have the red color incompletely dominant to the white color. And so instead of getting all red here, your heterozygous individuals are pink because the dominant can't fully mask the recessive trait. It partially masks it, and when you mix red with white, what do you get? You get pink. And so you get a very clear picture of what we would call incomplete dominance. Now, what does this look like? The blending theory, right? And so if you saw this, you're like, man, is this blending inheritance? But then if you do a cross between two F1 individuals, if it were truly blending, you would never get the red and the white back. But look what come back, our red and our white traits. So they didn't blend out, it's just when you have red and white together, you get a mix of the two. But they didn't blend out. It's not like they disappeared, the traits reappeared, but only in the homozygous individuals. Homozygous individuals for the red trait have red flowers. Homozygous individuals for the white trait have white flowers. Heterozygous individuals have a pink flower because the red can't completely dominate the white. Yeah, Levi.
So if the heterozygous kind of give us this mix, how do we know that one is dominant to the other?
It's not. I mean, you just use the language of incomplete dominance, and so you wouldn't say red is dominant to white. You could say red is incompletely dominant to white, but you could very easily say white is incompletely dominant to red. You could say either way. Yeah.
Is the color still being portrayed by only one gene? Like
Yes. Yeah, if you're going to get very predictable ratios like this, it is an indication that it's a single gene determining that color. Otherwise, your ratios would be more complicated. Because notice these are our genotype ratios from a monohybrid cross, right? Our genotypic ratios are 1 to 2 to 1. The difference is that our phenotypic ratios are not 3 to 1, like what we would have in a typical monohybrid cross, right? Because these two individuals, these two offspring, what phenotype would they have in a typical monohybrid cross? Well, what phenotype would they have? Not what are the ratios. A heterozygous individual, what phenotype would they have in a typical monohybrid cross? The dominant. But here they don't, because this red is not completely dominant to white. White is not completely dominant to red. You get a mix. So we actually, bless you, we end up with a third phenotype here. A phenotype that is only present in heterozygous individuals. Okay? And you're like, that's so cool. But Dr. Ingo, that wasn't the question. You asked, how does incomplete dominance differ from codominance? Okay? And so what do we need to talk about next? We need to talk about codominance. Okay? And the best example of codominance is in human blood type. There are reasons why pretty much everybody, when they learn these mechanisms, they learn the same examples, and it's because the snapdragon is the best example we have of incomplete dominance. Human blood type, best example we have of codominance. And so A is dominant to O, B is dominant to O, but A and B are codominant. And so what these all represent is they represent specific sugars on the surface of your red blood cells. So if you have type O blood, which happens to be recessive to all other blood types, but it also happens to be the most common blood type, you don't express any of these sugars on the surface of your red blood cell. You don't express the A sugar, you don't express the B sugar, and you certainly don't express both the A and the B sugar. You don't express either of those sugars. Doesn't mean that you don't express anything on your red blood cells, just means that you don't express these sugars. And this is why O is recessive to both A and B, because if you have A, you're going to put that A sugar on your red blood cells, right? So if you're putting the A sugar, that has to be dominant to not putting the sugar, right? Because if you put it, it's there. Yeah, sorry. Levi.
I was just wondering if O is recessive to the other blood types.
Yes, to all other blood
Why
types.
is it so common?
Because you just have fewer of those dominant alleles. So we did some stuff with Hardy-Weinberg last semester, right? In organismic. Okay, we didn't do anything with Hardy-Weinberg here. But so Hardy-Weinberg basically tells us that P is the proportion of all alleles that is the dominant allele. Q is the proportion of all alleles that is the recessive allele. And when we add those up together, that has to equal 1, right? Our proportions together have to equal the total. Well, if P, let's say P is 0.2. Okay? So let's say that's, actually, no, let's make it even rarer. Let's have some fun here. So let's say P is equal to 0.1. Okay? What would that mean Q is? 0.9. And we're like, well, cool. But what does that actually mean as far as what we would see in individuals? I'm glad you asked. The next part of the Hardy-Weinberg equilibrium gives us a way in which we can get genotype proportions, right? So we know that this is true for our allele frequencies. This is true for our genotype frequencies. So P squared is the proportion of the population that is homozygous dominant. Right? Right. 2PQ is the proportion of the population that is heterozygous. Q is the proportion of the population that is homozygous recessive. Okay? Well, if P is 0.1, what is P squared? 0.01. Okay? Right? So only 1% of your population is going to be homozygous dominant. Does it make sense? So 2PQ then is 2 times 0.1 times 0.9. 0.1 times 0.9 is 0.09 times 2 is 0.18. Do you agree? Okay. No? Somebody say no? There, I'm just hearing things. Yeah.
Sounded like your voice.
It was you? Sorry, on Mondays, I've come from a weekend of just having to tell my children no, like 15 times a day. 15 times an hour. And listening to them tell me no when I tell them something, so I just hear no's, it's magic. And then Q squared is 0.9 times 0.9, which is 0.81. And so the only way to get the recessive phenotype is here. This is the only way that that person's going to have the recessive phenotype, but it's still 81% of the population has that recessive phenotype. So that's what's going on with these blood types. So that even though these individuals would have, so if this is the A sugar, right? And you're like, well, then where did the B sugar go? We'll just say, well, very few people have the B sugar anyways, so we'll just assume that it's 0. I mean, you could do this with P plus Q plus R and have 3 alleles, but then the math is more complicated and I don't want to do that. So let's just say this is the A sugar, and the total number of alleles in the gene pool, it's just there are very few copies of that A sugar. And even though this person's going to have A-type blood, yes? This person's also going to have A-type blood because A is dominant to O, but that's only 19% of the population is going to have A-type blood, right? 81% has O-type, even though O is recessive to both. It's just because there's so many more of those O copies. And you're like, well, why is that? I don't know. I don't know what the ancestral condition is. You're like, was there a point at which there was only O-type blood? I don't know. Okay? But A and B are codominant. So if you have the gene or the allele for A sugar and the allele for B, you're going to put both on your red blood cells. You'll put A and B. And then it doesn't really matter whether you have one copy of the allele for A or 2 copies of the allele for A, you're going to put A sugar on your red blood cells. Are you going to put more? Maybe, but it doesn't matter. It doesn't really matter how much you have. Okay? Does that make sense? Okay. And then here's a little Punnett square where basically you put all 3 different options. So if you took just a random individual from the population, and you said, what might they have in their gametes? They could give the A sugar allele, they could give the B sugar allele, or they can give the O sugar allele. Okay? And they did I as lowercase to show that it's O, which is recessive to both. And then the capital I shows that it's dominant, but then they noted A and B, they're codominant. Okay? This is any random individual pulled from the population. They basically could give 3 different options in their gametes. Any random female pulled from the population could give 3. They give 3 different types of gametes, and then you cross these. Okay? And so if this sperm fertilizes this ovum, you end up with A-type blood. If this sperm fertilizes this ovum, you end up with AB blood. And then let me go down to here. If this sperm fertilizes this ovum, you still end up with A-type blood. Because A is dominant to O. Yeah, Peyton.
Would the offspring of that person that has the double dominant A or double dominant B have the ability to give a recessive?
So let me tell you this. If one of your parents has the double dominant, you can't have O-type blood, right? So I didn't know what my genotype was until, and I still don't know for sure, but my wife has O-type blood, I have A-type blood, and I didn't know if I have the double dominant or the recessive or heterozygous, but our kids all have O-type blood, which means that I'm heterozygous. And so my wife is O-type blood, all 3 of our kids have O-type blood. They lucked out. Because O-type blood, you can give your blood to anyone, but you can only take O-type blood. Because if you have anything on the red blood cells of blood that you're taking that you don't express, you're going to make antibodies against it. And so your question, though, was if one of your parents has the double dominant,
Well, I'm saying that if both parents had a double dominant, either A or B, and let's say I'm AB, does that still have the possibility of the person that I have kids with eventually also has the double dominant? Can there be O blood from that, or would it have to be
No, no. And so even if only one parent is a double dominant, you can't, your offspring can't have a O-type blood. Because you only have one option. If you're double dominant for the same allele, then all of your gametes, even if you were double dominant, you had A and B, all of your gametes are going to have either A or B. Yeah, that's a good question. That'd be very rare, because we know that I think it's somewhere around 45%. 45% of people have O-type blood. Maybe it's more. Maybe it's just, I think it's about 45%. You're going to look it up? Yeah. Micah's going to look it up for us. Okay, so to answer this question, well, of course it's going to be based on where you live, but we can get like an overall population. But you're absolutely right, because there is some genetic structuring. Okay, so the phenotypic ratios when you do a monohybrid cross with incomplete dominance, 1 to 2 to 1. Same as the genotype ratios of all monohybrid crosses. And that's because our heterozygous individuals have a different phenotype. They've got a mix of the two phenotypes. The dominant allele doesn't completely mask the recessive, although, like you were saying, Levi, can we even call it the dominant allele at all? I think you can make a good argument that either one is the dominant. Did you find?
O plus is 38.
O plus and 38 and 7 together is 45? Oh, man. Okay. Oh, wow. Even getting a standing ovation. Wow. I could not have anticipated that, but I did appreciate it. So both these traits disappear in the F1. When we do a one-character cross with two true breeding individuals, both the red and the white disappeared in F1, right? And you got that third phenotype. Notice this. The phenotypic ratios in a monohybrid cross with co-dominant, also 1 to 2 to 1, same as they are with incomplete dominance, right? And we're getting a new phenotype, right? And so this is like if we crossed a true breeding person with A-type blood and showed true breeding, all of their gametes will have the A sugar. With an individual that's true breeding for the B sugar, all of their gametes will have the B sugar. We're going to get a 1 to 2 to 1 ratio, right? 1 out of every 4 offspring will have A-type blood. 2 out of every 4 offspring will have AB type blood, and then 1 out of every 4 will have B type blood. Now, the differences are in the co-dominance, both alleles are obvious. They don't disappear. You see them both. It's not like with the Snapdragon where red and white disappeared and you got pink. Okay? It's A and B, they don't disappear. They're both there on those red blood cells. And so, which means that if somebody has AB blood, they can't give it to somebody with A blood or somebody with B blood. They can only give it to somebody with AB blood. That make sense? Because if you have AB blood and you try to give it to somebody with A blood, they don't have B. They're going to make antibodies against B. And guess what's on your red blood cells? B sugar. Okay? And so that's why we call AB the universal recipient, because they can take blood from anyone. They can take O-type blood, they can take A-type blood, they can take B-type blood, and then we call people with O-type blood the universal donor. They can give it to anyone because they're not putting any of those sugars. Yeah.
How does the positive and negatives come into effect?
Yeah, it's a separate factor that's also present. It's called the Rh factor.
Okay.
It's also present on the surface of red blood cells. So that's another one that comes in. Now this one is really common. Positive is dominant to negative, and the positive in that case is the most common. So most of the blood types are positive.
So if you had another 3-1 ratio with them.
Yeah, and so his numbers, he gave us 38% of the human population is O positive, 7% is O negative. So that's 38 to 7, that's greater than 5 times the number. So I mean, we could do some, we could do some numbers there and try to figure out the overall ratios, or we could just be like, okay, it's more common to have positive blood.
That's US.
That's US, yeah. which, I mean, the United States is a pretty good representation of the world as a whole, not of some groups. Yeah, there are some groups that are more isolated and who have some more unusual characters. All right, now, one thing you have to remember, and this is what we're going to deal with a little bit today, and then a lot on Wednesday and next Wednesday. And that's human genetics is really complicated because it's very rare to find a character that's influenced by only a single gene. Very rare to find a character that's influenced by only a single gene. And so then it gets really complicated, and then we don't do Punnett squares at all. Yeah.
So are any of these instances related to people with two different eye colors? Or like a dog you'd see?
So the two different eye colors is usually the result of a mutation that happens before those tissues split into two, because initially that the tissue that ends up developing into the iris is a single tissue, and then after they split, if you get a mutation in one line, usually it's a mutation in a single gene, and it's the gene responsible for depositing pigment, not for determining what kind of pigment you make, but for depositing it. And so a mutation in that gene makes it to where you can't deposit the pigment, so you end up getting a blue eye. And then so the other eye color is whatever eye color you should have genetically, but since you can't deposit the pigment in the other eye, then it just comes out as blue, and like a really, really light blue, because you literally have no pigment in there. Yeah. That's a good question. Did I answer your question? Okay. Because I said it was a good question, but I didn't know if I actually answered it. All right, so here's what I want you to do. We're going to take a little bit of a lecture break. And what I want you to do is this. I want you to practice a little bit with this codominant, and I want you to do this cross. Okay, go ahead and execute this cross. Work with those around you if you like. You work by yourself if you'd like. And give us all of the possibilities.
That's a good one. Can you? Oh wait, are you going?
Okay, so what term would we use to describe this genotype?
Oh, so wait.
This total genotype here, what term would we use? Heterozygous. What term would we use to describe this genotype? Heterozygous. It's like, well, this is kind of weird when you have more than two alleles, and it is. It's a little weird and it's a little bit more complicated. This is a heterozygous individual. This is a heterozygous individual. And we know heterozygous individuals at one locus, we call them monohybrids, and they can produce two different types of gametes. Okay? And so here we've got A and O, here we've got A and B. And so we'll do this cross, and we have A and O, and we have A and B. And so these two together will produce A-type blood, right? With a double dominant A-type blood. These two together will produce AB blood. Okay? These two together will produce A-type blood, but a heterozygous A-type blood, and then these two together will produce B-type blood. Okay? And so here's this codominance situation here, because when you do this, it would look as though that A-type blood would be dominant, because most of the offspring, half of them, I guess that's not most, half of the offspring are going to have A-type blood. But really, it's just because the other option is split into two different types of blood. Since A and B are codominant, 25% of the offspring will have AB, and then 25% of the offspring would have B-type blood, which wasn't present in either parent, by the way. Yeah, Chris.
So the offspring that has AB or AB type blood,
Yes,
is going to be the only offspring that has like the codominant alleles. Because
yes,
okay.
yep. Yeah, so what it would look like is 25% of the offspring are going to have this blood type, 50% of the offspring have this blood type, and then 25% of the offspring have a blood type that didn't exist in either parent. And you get kind of these weird ratios with these codominant alleles. Okay? All right. Now we're going to talk about what if we're dealing with more than one locus controlling a single phenotype. Okay? More than one locus controlling a single phenotype. And the first example of this is an answer to this question of what is epistasis? What is epistasis? And I'll show you. So here we have fur color and Labradors. And we have black labs, we have chocolate labs, and we have yellow labs or blonde labs. Okay? And so we've got three different options, and you're like, oh, this is going to be a single locus with three alleles. But it's not. It's two different loci, two different genes. And let me show you how it works. Okay? So here we take a true breeding adult for the black pigment, the black fur color, and a true breeding adult for the yellow hair color. That means all of the gametes produced by this parent would produce a black fur color. All of the gametes produced by this parent are going to produce a yellow fur color. Now, in the F1, all of the puppies are black. Okay? And so it tells you that black is dominant to yellow or whatever is going on here. And then so you're looking at here and then you're like, okay, well now we'll do our F2. And so we'll cross two individuals from F1. And you would predict if this were normal, if this were a normal dihybrid cross, we're going to end up with black and yellow. And that's what we're going to end up with. But then here we start getting some brown. Wow, this is really hard to see the difference between, here's a black lab, these are all black labs, and then these three right here are brown labs. And so what you find is that there's black is the capital B and is dominant to brown. Okay, so black is the capital B and is dominant to brown. The yellow comes when you don't deposit any pigment. It's not that there's a yellow pigment, it's that you're not depositing pigment. So you have a second gene that's called the epistatic gene that is controlling whether or not you deposit pigment. Because look at this. Here we have a yellow lab, but look at it, it's got two capital B's. You see that? It's got two capital B's, and so it should be a black lab. But it has two recessive alleles for the ability to deposit pigment. So it's making black pigment, but it can't deposit it. So you end up getting a yellow lab. Yeah, one sec. So here, this should also be black because capital B is dominant to lowercase b, to the brown color. But again, we can't deposit that pigment. Yeah.
So what happens to the pigment? Like, so it does, like, it makes.
Yeah, it accumulates in. So you have cells that are called. Oh man, it was right there. Chromatophores. You have cells that are called chromatophores that make pigment and then deposit it into epithelial cells. So it gets deposited into your skin and then it gets deposited into hair as well. And so that's why, have you ever noticed like a dog that's got like yellow and black patches? the skin underneath the black patches is brown, and the skin underneath the white patches is pink. And it's because that pigment's getting deposited in the skin and the fur. Right? Have you noticed that? You should do it. It's kind of cool. But here we've got, so here we have 3 out of the 16 offspring that are brown. And you have to have 2 lowercase b's to get that brown pigment, because lowercase b is brown and it's recessive to capital B, which is the black pigment. But you also need to have at least 1 capital E that allows you to deposit that pigment. Okay? Otherwise, so like if you look at this should be brown, right? 2 lowercase b's, this should be a brown Labrador. It should be a chocolate Labrador. But it's double recessive for the ability to deposit pigment. So it's making that brown pigment, but it's just staying in the chromatophores. It's not getting into the skin and into the fur. And the chromatophores are deep enough that you don't see them. Yeah. Okay. Does this make sense? So this is called epistasis. A good example of epistasis in humans, although it's not 2 different genes. There is one. Actually, I don't even know if. So anyway, sorry. I'll give you the example, and I don't know if either one of them are a single gene. But you have a widow's peak, right, where your hairline comes to a point in the middle, and then you have male pattern baldness. And so if you go bald and you lose your hairline, you may have a widow's peak, but it's no longer there, right? And so there you have the expression of another character, or another phenotype erasing another one. Yeah. And so the example when I mean, maybe we'll do it when we get into human genetics, is to show early pictures of Bruce Willis when he had his widow's peak, and then later pictures of Bruce Willis when he no longer has his widow's peak. I never had a widow's peak, so I can't do it for me, show you early pictures of myself before I started balding, but anyways. All right, and so epistasis. Basically what we have here is 2 different genes interacting. In a way, they're not behaving independently. In a way, they are violating Mendel's laws of independent assortment. It's not that they're being inherited as a unit, it's that one is completely masking the other. And so when 2 genes interact so that the alleles of one gene, we call that the epistatic gene, mask the effects of a second gene. We call that the hypostatic gene. And so with our Labrador example, what's the hypostatic gene? The one that gets masked. What's the hypostatic gene? Yeah, the pigment color. The gene for pigment color. And so what is the epistatic gene? The ability to deposit the pigment. Okay? So if you can't deposit pigment, it doesn't matter what color that pigment is. Okay? You get that yellow color. And then you might ask, well, why are they yellow if they're not depositing any pigment? It's just the way light gets reflected. Yeah, Levi.
So with the example of like male pattern baldness.
Yeah, male pattern baldness is certainly not a single gene.
Yeah.
But, sorry.
Would it still be considered epistatic even?
Yes, even if it's not a single gene. Yes. Yep.
Okay.
Yep. Then you would have to say like these suite of genes that might all cause male pattern baldness would all be epistatic to the hypostatic gene of a widow's peak. And I don't even know if a widow's peak is determined by a single gene or not. So yeah, genes that determine whether or not pigment is deposited are oftentimes epistatic. We see this not only in Labradors, we also see it in some breeds of horses. Yeah.
So does epistatic refer then to things that are expressed at birth and then things that can also be acquired later on? Because,
Oh, are you getting into, you might be thinking epigenetic.
well, I was thinking because a dog is born with a certain fur color.
Yeah.
But baldness happens over time.
Yeah, so epistasis is, it's kind of independent, it's whatever's going on right now. So like it could be something that happens from birth where you always mask that, or it could be something that happens later on. Either way, it would still fall under epistasis. Yeah. So the answer to your question, I think, is yes, if I remember your question correctly. So, all right. Any other questions? We have one more example of some things that don't fit Mendelian principles, and here it is. And the question here is, why do some characters form a continuum? So Actually, no, we may have a second. Sorry. Why do some character traits form a spectrum? And so a good example of this, I don't know, you tell me, what's a good example of a phenotype or of a character that occurs over a spectrum? It's not discrete like brown pigment, black pigment, yellow pigment. Like it's over a continuum. Yeah, Davies.
That would be eye.
What's that? Height? Yeah, height. Human height is a great example of this. And here's the image. So this is a statistics course at BYU. And here they have the statistics course grouped according to height. And so we've got the shortest individual, two individuals tied for the tallest individual, and then you have a nice bell-shaped curve here. Okay, where we've got a continuum. Here's the actual data from the class, range of heights, from the shortest individual to the tallest individual, and these numbers represent the number of individuals at that particular height. And so you get somewhat of a nice bell-shaped curve here, where you have a continuous distribution of those phenotypes. And so here's forcing it to fit a bell, cleaning it up a little bit. And the question is, well, why does this happen? Why does this happen? Why isn't it as simple as widow's peak, yes or no? Hitchhiker's thumb, yes or no? Right? Why isn't it that simple? It's like, how do you have I guess there is kind of a spectrum, because a lot of people have hitchhiker's thumb, but it's not like this. I mean, that's 90 degrees. And it still goes back to normal. I mean, it's impressive, I know. Wow, sorry, I got myself all lost. And so the answer to this is whenever you have multiple genes influencing a single character, we call this polygenic inheritance. What does poly mean? Many or multiple genic genes. And so whenever you've got multiple genes that are influencing the same character, you're going to tend to get a continuous distribution of phenotypes. And so this is even true for eye color. And so we treat human eye color as though it's a discrete character, where it's like you've got blue, you've got brown, you've got green, you've got hazel. But if you actually look at people's eye color, you realize it's more of a continuum, right? You have some things that it's like you default to saying that it's brown, but when you look closely, it's not brown. Okay? It's kind of something a little bit different. And you're like, well, they just kind of lump everything under hazel, right? And then just treat it as its own category. So we've got several genes contributing to the phenotype, and typically is going to form a nice bell-shaped curve where most of the individuals have whatever the average condition is, the mean or the average condition. So another big thing to note when you've got this continuous distribution and you've got these polygenic characters is that the environment is going to play a large role in the expression of those phenotypes. And if you've got a single gene, usually the environment plays a very small role. It's like you've got a single gene, and that gene is going to determine most of the character. But when you've got multiple genes, your environment is going to impact how much those genes get expressed. And you're going to start to allow the environment to play a larger role. So human height, we've been using this as an example. 20% of human height is determined by environmental factors. 80% is determined by genetic factors. 20% is determined by environmental factors. So you could be genetically predisposed to being tall, but not get the proper nutrition necessary to actually make that happen. Right? Or you could be genetically predisposed to be short, but you were exposed to growth hormones and you ended up growing taller than what you should have. This is what we do with our chickens, right? So you can buy a chicken that's 5 pounds for a single chicken instead of what would be normal, like 3.5 pounds, right? So you give it growth hormones. It's not genetically predisposed to be a massive chicken, but it becomes that when you give it growth hormones. You trick it into being bigger than what it should be. And so we call these characters We call these characters quantitative traits. And so the other characters, where they occur in discrete categories, we call those qualitative traits. Quantitative traits are those that vary over a continuum, and those that have discrete categories, like our Labrador example, where you have black labs, brown labs, or yellow labs. Those are discrete traits. Okay, we do have one more question. Sorry, I told you that was the last one. I got excited. There's one more. Any questions about this? Okay, the last question is this one. Are there genes that impact multiple phenotypes? Because we just talked about an example where we have multiple genes impacting a single phenotype. Well, what about, do we have a single gene that impacts multiple phenotypes, right? Because like height is a single phenotype impacted by multiple genes. But what about a single gene that impacts multiple phenotypes? Great example of this, sickle cell anemia. And so we've talked a little bit about sickle cell anemia in this class already. And so we have a characteristic in which a lot of our genetic illnesses are what we call pleiotropic, where you've got a single mutation. Cystic fibrosis would be another good example, where you've got a single mutation contributes to a genetic illness or determines the presence of the genetic illness, but there are a number of different phenotypic results of that single mutation. So genetic illnesses are usually pleiotropic, like sickle cell anemia. And so with sickle cell anemia, you're going to have blood clotting issues. You're going to have issues in tissues that are highly oxygen dependent because you can't carry oxygen as efficiently. So you're going to have neurological issues. You're going to have blood clotting issues. You're going to have issues in other tissues that are oxygen hungry, muscles, the liver. Okay, a lot of these oxygen hungry tissues are going to be impacted by a single mutation. Okay, and so we've got human height, multiple genes, single phenotype, pleiotropy. Okay, single gene, multiple phenotypes impacted. And again, your monogenic genetic illnesses, meaning genetic illnesses caused by a single gene, most of those are going to be pleiotropic. All right? Any questions about this or anything else? Does it make sense how these don't really agree with normal Mendelian ratios? Yeah, Levi.
My question was just a pleiotropic genetic, problem. Is it like expressed in different plants and different phenotypes and different plots?
It could be that mutation results in different proteins in different tissues, or it could be that single protein is involved in a number of different tissues in basically the same format. And so with sickle cell anemia, the protein that's malfunctioned is one of the proteins in hemoglobin. And so it doesn't carry oxygen as efficiently, and it tends to cluster up and create a sickle-celled red blood cell. With cystic fibrosis, it's a protein that moves ions. And so if you can't move ions efficiently, you can't get water to move efficiently. And so you get, body fluids that are too thick or too thin, and you've got issues expelling metabolic waste. But it's a single protein. It's just a protein that's expressed in a number of different tissues.
Would that also be a pleiotropic?
Yes. Mm-hmm. Yep. Absolutely. All right. Well, for those of you that have lab this afternoon, I will see you at 1:55. For those of you that have lab on Thursday, I will see you Wednesday morning at 11:45. Yeah.